thanks to all of you who came on time. We're going to give it another 2 minutes. And then we'll get started.
Sorry.
No.
Did you?
Oh,
She
Hey, Eric. Done.
Okay.
I'll work on that.
All right, so we're gonna get started. Let me welcome all of you. This is the final seminar in the 2014-2015 academic year series of our seminars on ethical, legal, and social implications of genetics. We will pick up again in September, and we will get notices out to all of you if you're on our email list about that. And if you're not on our email list and would like to be, please just jot down your name and email and give it to me before you leave today, and I will be more than happy to add you to the list. Also, for those of you who might have thoughts about people who you would like to see up here at the podium next year, people who are involved in ethical, legal, social studies of genetics or involved in genetics more broadly and have reflected or thought about or written about these broader issues, please let me know. You can tell me at the end of this session. You can send me an email if something occurs to you afterwards. We will be planning next year's series in starting in just a couple of weeks in July, and your input is very much valued.
You meet every 2 weeks?
Every month.
Every month.
Right. So for our ultimate presentation of the academic year, we are really delighted to have Eric Turkheimer with us from the University of Virginia. Eric is one of the deans of behavioral genetics in this country, a former president of the Society for Behavioral Genetics, and someone who really has enormous stature in the field, and that stature comes not just from his studies in behavioral genetics, twin and other models of work, but also from the ability that I think will be on display for all of us here this afternoon, which is the ability to step back from the data and ask what the bigger picture is all about and what the broader implications of the data are, and how really to understand both the potentials and the limits of the field. So we are delighted to have Eric here with us this afternoon. Thank you.
Thank you, Paul. It's really nice to be here. And thank you all for showing up. This is where I'm from, very late in the academic year to be giving a talk. I think if in the psychology department somebody came and gave a talk in June, he would be talking to himself. So it's nice as summer starts for you to be here. And I am going to speak about theory today. It's unusual for me. I don't think I have any data slides of my own in here. Bear with me while I do that. Thank you, Paul, for referring to me as a dean of behavior genetics, though I must say that makes me feel a little old. And one of the things that happens to you when you've been in the field for a long, long while is you start to think about things that you said yourself a long time ago. And the topic today, what I'm going to talk about, is the notion of there being individual genes. That is not kind of heritability, twin and adoption study genetics of things, but individual bits of DNA that are related to very complex behavior. This is something I've thought about for a very long while. And a long time ago, before the Genome Project was completed, in a section of a paper called Reasonable Expectations for the Genome Project, I said that such associations are real and potentially interesting, but they remain correlations and small ones, not evidence of substantial causal pathways between individual alleles and complex behavior, or evidence of genes, quote, for extroversion or intelligence, or evidence that future scientific efforts will be most productively applied at a genetic level of analysis. That's almost 20 years ago, and I think the point of view that I took back then has been fairly well borne out by what's happened in 17 years since, and that's really what I want to talk about. I'm going to frame my comments in terms of thinking about a paper that was published in Science a couple of years ago in 2013. I don't know if you saw it. It has a title, GWAS. GWAS, and I'm going to say more about it. I presume you know in general that GWAS is a genome-wide association scan of 126,000 individuals, identifies genetic variants associated with, of all things, educational attainment. So somebody found a very large sample of people on whom educational attainment had been recorded, searched through about 1 million SNPs on a SNP chip, and identified 3, in this case, variants that were significantly associated with educational attainment at p less than 10 to the minus 8, which is the current genomic standard for significance. Got it into Science. That's the author list. So there was close to 125,000 authors. And I don't, probably there must be Columbia people on that list. I don't know if anybody's here. But I put it up by way of saying that my message today is not that that study is wrong or badly done. I think what they found is true. I do think those 3 SNPs are probably associated with educational attainment. My criticism is not on that level. What I think is that it doesn't matter very much, and that it's ultimately not surprising, and that it's not, in fact, the direction of the field of behavior genetics is probably going to evolve over time. That's just more as the study itself and the 3 SNPs that were significant. To think about what this study means, you have to back up in behavior genetics to an older concept in behavior genetics, and what many of you probably think of when you think of what it is that behavior geneticists do, which is the concept of heritability. And I've really skipped over most of my "this is the way twin study works" slides that I sometimes include because I get bored with them myself. But in behavioral genetics, you can study the educational attainment of identical twins and fraternal twins and show that identical twins are more similar in their educational attainment than fraternal twins are. And that leads you to a construct that is called heritability. It's an index of the fact that educational attainment is not independent of genetic differences among people. And 30 or 40 years ago, when I was getting started in the field, behavior genetics was very much about the computation of the heritabilities of things. Once people figured out how to do basic twin studies, there were an obvious set of questions about behavior to ask whether identical twins were more similar than fraternal twins. The method itself goes back to Sir Francis Galton in the 19th century. Galton was interested in what he called eminence, which is a construct that evolved into the construct of human intelligence, and people did famous studies of the heritability of human intelligence, personality, which this slide happens to be about, of course, psychiatric genetics, schizophrenia, depression, and what behavior genetics used to do was to compute these heritabilities. It's a variance component that varies from 0 to 1, and this is a column of heritabilities, all of them as they tend to be pretty close to 50%, with standard errors next to them and little a's and b's telling us whether they're p less than 0.05, and lo and behold, they're significantly different from 0. In fact, a funny thing happened when people started computing heritabilities. People thought at the time that behavior genetics was going to offer us this interesting tool that was going to allow us to divide the world up in general, and in particular the world of human behavior up, into some traits that were very heritable and therefore, quote, genetic, and other traits that were less heritable and maybe more whatever the opposite of genetic is, environmental or psychological or something like that. Well, it didn't happen. Interestingly, what happened, and this is where I first came into the field, and I would sit as a graduate student in the back of seminar rooms when people said, oh, well, we undertook a twin study of X. And I would say, okay, I'm going to write down what happened in this twin study before they report the results. And what happened is everything is heritable. Identical twins are more similar to each other than fraternal twins are for everything. So once people had done the twin studies of intelligence and personality and schizophrenia, there were still a lot of dissertations left to be done. There were a lot of twins around waiting to participate in twin studies. So people did twin studies of whatever. This table is from a twin study of how much TV kids watch. The heritability is for 3 different versions of it. No, it's 3 different ages, or 0.5, 0.6, and 0.2, with fairly large standard errors on them. So if you just said they're 0.4, you wouldn't be wrong. And this is true of everything. How often you go to church is heritable. Your political attitudes are heritable. The kind of food you like to eat is heritable. Everything is heritable. And the reason everything is heritable is not because twin studies are wrong and they're giving this false impression of universal genetic influence. In fact, twin studies are right. And our genes and differences in our genotype do, in a very general way, influence everything we are and everything we become. That, I think, is sort of obvious to many of us now. It's certainly obvious to me. But it's important to remember that there was a time that that wasn't so obvious. And there was a time when a behavioral geneticist giving a talk like this would have demonstrators out front because people were shocked by the idea that genes might have anything at all to do with differences in behavior. But the fact that heritability is universal places limitations on the conclusions we can draw from discovering that something is heritable. In particular, once we realize that television watching is heritable, we have to recognize that the fact that something is heritable does not imply that that something has a genetic mechanism. In fact, twin studies are based on a very, very non-mechanistic model. I can't get into a lot of the detail here. But twin studies, like most human statistics, are based on Gaussian distributions, normally distributed things. Genes themselves, of course, are discrete things. And the theory that underlines twin studies says that if we're interested in the genetics of height, for example, that there are thousands and thousands of genes that influence height. Right? And all those coins get tossed. And the distribution of number of tall genes then forms a normal distribution as a Bernoulli process that then leads to the normal distribution of height. So the statistics that underlie twin studies specifically denies that there are genes for things. That very early on in the development of behavior genetics led to a crisis that was given up on too soon in the way people think about behavior genetics. John Paul Scott and John Fuller and what's his first name? Fuller and Thompson, the other early textbook. The early work in behavior genetics was largely experimental. It was largely done on temperament in dogs. And these were people who were not interested in human behavior. They were interested in genetic and environmental mechanisms that are very biologically oriented that led to temperamental differences in dogs. And these people who were, John Fuller was the first president of the Behavior Genetics Association, was very, very concerned about human twin studies because he didn't believe in the etiological relevance of polygenic models. So in the part that's bolded there, he said, thus, if most behavior traits must be fit to polygenic models, we may be left only with statistical analyses of such problems as how many genes are involved in the specification of almost infinite number of interactions between them. Such mathematical exercises seem to us to have only trivial importance and furthermore to be of small interest to most biologists and psychologists. Probably the greatest clinical psychologist of the last century, Paul Meehl of the University of Minnesota, who studied, among other things, schizophrenia, and for his entire career harbored an obviously false single gene model of schizophrenia, did so. I think the reason he favored an obviously false single gene model is because it didn't seem possible to him that polygenic twin models could actually be biologically true. He said one understands fairly clearly what it means to conjecture that a big effect monogene is the specific etiology of a disease. But once we've excluded that simple situation, the very meaning of the phrase specific etiology begins to fuzz up. In the sense that it's hard to know what we mean when we say our genes contribute to how much TV we watch. A couple of other points to go over relatively quickly. Sort of the next thing that behavior genetics thought it was going to be about, once it became clear that everything was heritable, was maybe some things are more heritable than others. And maybe we can learn something about behavior by discovering some behaviors that are highly heritable and some behaviors that are less so. Long story short, the answer is no. This is from my mentor, John Lolan, who studied the genetics studies. He's still with us, studied the genetics of personality, and the slides from a talk I give on the genetics of personality. And what Lolan, over a 50-year career, has shown is that all personality traits are heritable, and they're all about equally heritable. You really can't make a science out of whether neuroticism is more heritable than extroversion. It goes back and forth, which is really the next slide. That, in fact, whether something is more heritable or less heritable than something else isn't a consistent property of traits. Again, from the personality literature, this was another crisis back in the 70s. They started doing meta-analyses of whether the heritability of a trait in study 1 predicted the heritability of the trait in study 2 relative to other traits, and it didn't. The correlation was 0 across studies. Things are heritable about 0.5. It goes up and down with the studies. Another idea about what the universal genetics of behavior might mean is that things that are heritable, that once we know, so we work in the behavioral sciences in a lot of hypothetical constructs. And it's very hard sometimes, extroversion, to know exactly where our hypothetical constructs are coming from. And one impulse that people often have is to think, well, if something has a genetic basis, that must mean it's real. A famous quote from the psychiatric geneticist Seymour Ketty, who was replying to Thomas Zaz at the time, and the idea that mental illness is a myth, said, well, if schizophrenia is a myth, it's a myth with a strong genetic component. But that doesn't work either. Because not only is everything that one might think of to measure heritable, if you make up things to measure, it's heritable. So this is Wendy Johnson, wrote a paper about the heritability of personality. She gave a big complex of personality scales to a big bunch of twins, computed the heritability of all the traits. This is the Minnesota Personality Questionnaire. Down the left, well-being, social potency, and they're all MZ higher than DZ. They're all heritable. But then she made up traits at random. She picked 20 items from the scale and put them together and called them, junk trait 1, junk trait 2, and they're all heritable. Moreover, getting closer to the point of the talk, as time went by, one thing that people thought is that heritability would... I forget if I have the particular slide in here. We're now in the 90s. Okay, people have been doing twin studies for 50 years. Figured out, demonstrated that everything is heritable, that nothing is independent of genetics. But twin studies, of course, for the biologically oriented can be very frustrating because twin studies are done at a great remove from any actual DNA, which at the time we didn't have. So in the 90s, everybody was anticipating the completion of the genome project. And the question on everybody's lips was, what's going to happen when we have the actual DNA? I mean, that's really the topic of this talk. And what people thought was going to happen, or hoped, was that twin studies were going to be the guide to where molecular genetics would be especially productive. So you find traits that are highly heritable, and that's where the gold is. And so you dig there. Those are just quotes of people singing that. And what's interesting is that it didn't work. This is one of my favorite papers from Neil Reisch, showing that in cancer genetics, the cancers that are the most heritable are the cancers for which they've had the least luck finding any actual genes. And now, whereas the cancers that are the least heritable are the ones for which they have found big genes. And in fact, that makes perfect sense based on that first slide I showed you with the normal distribution. Highly heritable cancers are common cancers against polygenic backgrounds. And those cancers are well represented by twin studies. And there is a general background genetic risk to those kinds of cancers, exactly like there is to being extroverted. Other kinds of rare cancers are contributed to by rare single genes of large effect. And those kinds of cancers are not, they're genetic, but they're not heritable because those genes do not vary widely in the population. Bottom line message, and something I like people to take away from my talk, is that, in fact, traits don't have heritabilities. I think the most common misperception of behavior genetics is that it's about, people will ask me all the time, well, Eric, you're a behavior geneticist, tell me, what do you think the heritability of intelligence, that's the favorite, or schizophrenia, depending on where I am, is? And the answer is it's a bad question. It's not zero. Genes are important for these things, A. B, it depends on how you do the study and how things vary. The famous example of this is, not mine, is that the heritability of having 2 arms is 0. Why is that? Well, the reason for that is that whatever the genes are that are involved in our growing 2 arms, they don't vary among any of us. It's constant. Right? So genetic variation has no relationship with degree of armedness. And in fact, to the extent that some people unfortunately have fewer than 2 arms, it's usually because they were in an accident. So arm variation is largely environmental. We would not want to conclude on that basis that growing arms is not a genetic process. Of course it is. Heritability is about variation, and variation changes from situation to situation. Sort of section 2 of the talk here. Trying to understand the problem of how it is that genes have their universal effect on behavior. We can learn a lot from a related question. I'm not a psychologist. of thinking about how environment obviously has a universal effect on behavior. How does environmental influence work? And though the story is in many ways discouraging, we know a whole lot about that. And I think we have a much more sensible set of expectations about how environmental influence works. Suppose you're interested in a classical social science question. What are the effects of divorce on kids? That's a very hard thing to study. And the reason it's a hard thing to study, I was just saying this upstairs, is that what you want to do, if you want to be a scientist of divorce, is you want to randomly assign families to divorced and non-divorced conditions and see what happens to their kids. You can't do that. And because you can't do that, you have to resort to other means. And the old-fashioned way of doing this in psychology and sociology, and it's remarkable how much of this there was say, okay, we're interested in what the effect of divorce on kids is. Here's how we're going to do it. We're going to find some divorced families and some non-divorced families. We're going to look at their kids, identify some differences between their kids, and voila, there you go. There's the effect of divorce on kids. Now, when I tell the story that way, hopefully it's obvious that that methodology is really lame. And the reason it's really lame is exactly that you can't randomly assign people to divorce conditions. So divorced families are different from non-divorced families in myriad ways, right? Not just that they're divorced. They're poorer. They might be different genetically. So divorce is a very complicated causal process that's very difficult to understand. And just doing elementary social science here, so we're interested in the pathway on the bottom, the relationship between parental divorce and substance abuse, say an outcome I picked, substance abuse in kids. We wonder if that's true. Well, we can't just look at the correlation and conclude that it's causal because there are possible confounds of that relationship, right? It could be that there's a genetic predisposition to substance abuse that is related to the genetic background that might lead people to get divorced. And that predisposition that then gets passed along to the children and induces a pathway that goes that way through the genetic predisposition and is non-causal. I mean, people who do social science are used to this kind of thing. The same thing could be true environmentally. It could be that poor people are more likely to get divorced. Poor people live in neighborhoods where there's more drugs around, and therefore their kids are more likely to use substances. So it winds up that divorce and substance use are correlated, but it's a completely non-causal relationship. So we're left in the kind of classical multiple regression kind of situation, that we have a purported pathway between parental divorce and substance abuse in kids, and somehow we want to control for the genetic and environmental compounds of that relationship. This process, this being in this particular boat, is my definition of social science. Social science is an attempt to do science under three conditions. There's a large, complex set of potential causes, like the effects of divorce on kids, or looked at the other way, why do some kids start using drugs? Those potential causes are all non-independent. That is to say, we can't randomly assign, really. So poverty as a cause of problems in kids and divorce as a source of problem in kids are correlated with each other. And it's very, very difficult to break those things up. So what do you do as a social scientist when you're faced with that? Well, basically you do statistics. And those of you who spend any time doing that know that it's doing statistics to try and get causal inference out of non-experimental data is interesting on the one hand and sort of hopeless on the other. And this is actually from another talk. The only one of these various things that I'm going to talk about is the first one, statistical significance, because that's very important in the Genome Project. I am a big opponent of p less than 0.05 significance testing. Those of you in related fields know that that's, by now, a very commonplace point of view. So p less than 0.05 means that given a null hypothesis of no association, what's the probability of drawing our observed data? There are enormous problems with doing that I won't get to other things. That very relevant to what we're going to talk about is that our ability to reject that null hypothesis always depends on the sample size. Many people, it's been said many times that a p level is an indirect measure of how big your sample is. And if your sample is big enough, p is going to be less than 0.05, guaranteed every time. Anyway, interesting to note that in many high-quality social science journals nowadays, if you report your result in terms of p less than 0.05, they'll send it back to you without review. Then we'll contrast that with some other things. NHST, null hypothesis significant testing, is a way of discriminating actual associations from those occurring because of sampling error. It has nothing whatsoever to do with causation. It tells you nothing about whether parental divorce is really causing drug abuse in kids. It's only about sampling theory. So this social science paradigm, which is related to something called the non-shared environment, is the ultimate paradigm of social science. The non-shared environment is a behavior genetic term, which, by skipping over those early twin slides, I'm going to have to describe to you quite quickly. The non-shared environment is a variance component like heritability that comes out of twin studies that is the error term in the study. It refers to everything that makes monozygotic twins reared together different from each other. So MZ twins are similar in personality, but they're not identical. They share all their genes. They share a family environment. Well, whatever is left over that makes them different from each other is called the non-shared environment. And 20 years ago, Robert Plomin, the leading behavior geneticist, noticed that most of the environment, almost all of the environmental variance in twin studies is attributable to the non-shared environment rather than the opposite of it, the shared environment. The shared environment is what you think of as the environment, your parents and your families and your socioeconomic status and where you live, because kids raised in the same family share all those things. And it's very hard to show that those things have any effect at all. What seems to be important are the things, the environmental things that make kids raised in the same family different. And they identified a famous three-step research program. Number one, quantify the magnitude of non-shared environmental effects. Number two, identify specific non-shared environmental, within family environmental variables. And three, study the causal relationships between within family environment and behavior. So this proposal is to try and identify what parents do to make their kids different from each other. So if parents treat kid 1 different from kid 2, does that make kid 1 turn out different from kid 2? This is just a mock-up of the kind of study that gets done. We've actually done this very study about divorce. So one way you can study divorce in twins is to find twin parents, not twin children, twin parents who are discordant for divorce. One identical twin is divorced and the other is not. And then their children become an interesting comparison group for each other. If the children of the divorced twin are using drugs, well, then it's an interesting question to ask, what are the children of the non-divorced twin doing? And I won't spend a lot of time on the logic of that, but that's the way these studies work. And I did that's one of my few actual data slides here. Mary Waldron and I did a meta-analysis of all of the within-family studies that were done under this rubric. And what's interesting about them is that when you study specific measured environmental variables, the effect of the non-shared environment dries up and blows away. You can't identify anything that parents do differently with their two children that make their kids different from each other. How they talk to them, how they treat them, the nature of their relationship. In general, something makes kids different, but when you try and break it up into individual environmental chunks, you can't find it. You might call this, anticipating where we're headed here, the missing environment problem. Something makes children raised in the same family different from each other. It's easy to show. But when we try and break down what that something is into individual causal environmental events, we can't find it. The truth is, this I don't really think is much of a mystery when we talk about the environment, because we've been doing social science for so long that we're used to it. Long ago, people had very simplistic models of how the environment, this is also from my personality talk, of how environmental causation might work. I don't mean to make fun of psychoanalytic explanation here. I was saying I have leanings that way myself. But I don't think anybody thinks in a simplistic way anymore that how your toilet train determines your personality. That there's this big environmental cause and it has a big behavioral outcome. In fact, whoops, messed around with my slide a little bit. In fact, we know that there are a lot of environmental events that influence personality. And this is meant to kind of model a kind of multiple regression additive model of how a bunch of different environmental differences might all add up together and produce personality. And you can do a multiple regression study that way, but in fact, we know that environmental causation doesn't really work that way either. Environmental causation, in fact, works much more like this, that all of the environmental events are inputs into a developmental process. This is a kid growing up, and this is all the complicated things that happen to the kid as he grows up. And out the other end, somewhere, comes that kid's phenotype, that kid's observable behavior. And it's very difficult to trace the individual pathways of environmental causes again through that developmental thicket. You can still detect correlations across it. There's a lot to be said about that. And this idea in social science environmental causation is something I didn't call, Robert Pullman called it the gloomy prospect, but he called it the gloomy prospect by way of saying he couldn't imagine anything so horrible would happen. That it might turn out that the way development works is that the causes underlying our development are a tangle of mostly random, tiny, individual processes that are very, very difficult to study causally. And that was a gloomy prospect that couldn't possibly be true. Well, I'm here to say the gloomy prospect is true. And that's why it's so frustrating to be a social scientist. So anyway, where are we? Traditional social science involves the search for cause under conditions of multiple correlated predictors and absence of experimental control. P less than 0.05 doesn't help. You can do statistics to try and help, and there are some things you can do, but mostly you can't. Family-based methods work pretty well, studying differences between identical twins, but when you do that, most of your purported causal effects go away. And as a result of all this, we no longer really expect behavior to have mechanistic environmental causes. If I say to you, why do some kids become problematic drug users and others not? Nobody thinks that somebody's going to make a discovery, it's lead paint. That we know that that's not true. That however it is that environmental causation works, it's not that way. So, onto the genome. My takeaway message is that the best scientific model for the molecular genetics of complex behavior comes from what has already happened in the non-shared environment. And if you take that Plowman and Daniels 3-step research program for the non-shared environment, where we had a big variance component that had been named the non-shared environment and we wanted to figure out how it works, you can transform it. First you quantify heritability, and everything is heritable. Then you identify specific UTLs or SNPs or genes that underlie that heritability. Then you try and find the causal associations between, why does that still say it's a mistake, between those genes and behavior. Oh, there we go. And that's the Human Genome Project. The Molecular Human Genome Project is about trying to find the individual pieces of DNA that constitute the variance component that had been identified by the twin studies. Now, a 2-minute history of the molecular genetics of complex behavior. It started out with linkage studies, which are a way within families of showing a relationship between linkage disequilibrium in the genome and differences within families in outcome. It was actually done before the genome project was completed. It was the first thing that people tried when they thought they were going to find the genes for schizophrenia. I love that quote from Tim Crow. Many in linkage research thought that success was inevitable. One would drain the pond dry and there would be the genes. And he's saying that by way of it didn't happen. There was a time in the 90s when you couldn't open a copy of Science or Nature without seeing another linkage study for major psychopathology, mostly. And famously, they didn't replicate. The next, not quite sequentially next, thing that was tried is what's called candidate gene association studies. Candidate gene association studies are extremely simple. You pick a gene of interest and see if it's correlated with your outcome of interest. That's really about it. You have more statistical power because you don't need sibling pairs to do candidate gene association studies. And now around 2000 or so, you couldn't open Nature Genetics without seeing a candidate gene association study for schizophrenia or bipolar disorder, and now intelligence and extroversion and violence. And just as famously, those didn't replicate either. That's a very interesting paper about candidate gene studies for intelligence, making a very hard to argue with statistical argument that whatever was reported for genetic associations with, in this case, intelligence, were almost certainly false positives. Because it's known that the effect sizes are very small and these studies were all radically underpowered. It's hard to overemphasize what a crisis the failure of linkage and association studies created in behavior genetics broadly, psychiatric genetics and psychological genetics. Because people really thought that we were going to drain the pond dry and find the genes. And for 15 years in, there still to be no identified genes related to schizophrenia was an enormous crisis in the field. And genome-wide association studies were something that were developed specifically in response to this crisis. And I don't have time to go into a lot of detail about GWAS, but what's interesting about it is that it's the very opposite of the way most of us learn to do research. When I learned to do research, I was told I was supposed to have a hypothesis, and I was supposed to have a theory, right? And what the failure of candidate gene association studies taught us was that theories are extremely dangerous. Because theories bias the things that we look for and get us to look in places where we think we're most likely to find something. And under conditions like that, most reported findings are false positives. So anyway, the way GWAS works is there are now SNP chips it's gone from a few 100,000 to now about 1.5 million SNPs, single nucleotide polymorphisms, which are individual units of DNA, can be measured on them very cheaply. It's quite easy to give them to at least tens of thousands, if not hundreds of thousands of people, and it produces then 1 million significance tests, whether each individual SNP varies between schizophrenics and non-schizophrenics. And of course, which is amazing, but of course the problem is that no matter how you do it, you're doing 1 million tests. Which leads to some fairly severe significance problems, which has been, and the response of the field has been to require very, very rigorous significance, p less than 10 to the minus 8, in order to accept it as real. These slides are a little out of date, and I'm running out of time, so I'm going to go fast. One thing people thought they would try is height. Certainly this would work for height. And it sort of does. You can, in 63,000 people, identify a few SNPs that are related to height, and that's where this is out of date. It's a little more than a few now. They're probably up to 50 or 60 SNPs now that they've identified that are significantly related to height. But one particular thing interests me here. that they talk about all their significance testing and say at the bottom, this means that because they're very significant, associations are likely to reflect true biological effects on height. And the question is, what's a true biological effect? We're in the social science situation here. We have many potential causes with low correlations with outcome. The potential causes are all correlated with each other. How can we tell which is real? And the answer is they do very rigorous significance testing, which, I'm going too fast here, systematically answers the wrong question. Remember what we decided when we were talking about divorce effects on outcome in kids. Significance tests are about sampling error. Significance tests tell us how likely are these associations to have occurred because of sampling error. They do not tell us how likely are these effects to have occurred because of identifiable biological reasons. Well, that's what I just said. The irony in all this, which I've really already said, is that in traditional social science, in the Journal of Abnormal Psychology, you can't report a p-value anymore. Yet, in the Genome Project, at the very outer limits of what our genomic technology allows us to do, the science has become one long, extended exercise in p-level testing. And well, I just think that's very ironic. And I don't think there's good reason to think that a methodology that has failed so miserably in sociology is all of a sudden going to start working in genomics. I hate to have to, I hate it when people skip slides, but I think I'm going to have to do a little of that. Can I go pretty close to 4 o'clock? That way nobody gets to ask any questions.
It's all straightforward.
And interested, one of the classic problems in GWAS is technically called population stratification, and is commonly known as the chopstick gene problem after somebody named Turkheimer. The chopstick gene problem goes like this. You want to find a gene that causes people to use chopsticks. Here's how. Get a big GWAS sample that is half American people and half Japanese people. Then search through the genome and find whatever gene you want that is more frequent in Japanese than it is in Americans. That gene then in your sample is by definition correlated with chopstick use. So there you go, there's your chopstick gene. That's called population stratification. It's viewed as kind of a methodological, technical problem in genomics. And as a result, people try and get samples that are very ethnically homogeneous, which is, of course, not something you can do perfectly. There are a lot of statistical methods to deal with it. But my point is that that problem is, in fact, it's a social science problem. It's a problem of inferring causation from correlation. It's exactly the same problem that we deal with when we're trying to understand the relationship between parental divorce and drug use in kids. There's a gene that's associated with use of chopsticks. You want to know if that relation's causal, but it might, in fact, root through Japanese cultural ancestry. It might be that in the same way that a gene for marital discord is actually rooted through a gene for alcoholism. Moreover, in genomics, 2 concepts are particularly important: polygenicity and pleiotropy. Polygenicity refers to the fact that anything that we're interested in, that's the first thing I said, is related to many, many genes. There is no gene for anything that we care about behaviorally. Right? So we have genes on the left, outcomes on the right, and many genes produce any given outcome. Pleiotropy refers to the fact that any given gene has multiple effects. All of which is by way of saying that genomic causation is no different than social scientific causation. It's highly complicated. And the exact same things are true of genomic causation that are true of environmental causation. the oh God model, the one gene, one disorder model, is that's actually a thing that people used to talk about, the oh God model. And that's the way people first thought about the genesis of schizophrenia, that there was a single major locus. That micro ocean is referring to personality. That's the 5-factor model of personality. That maybe there's a single gene and we know that's not true now. We can go from there to, this is a SNP model, an additive SNP model, that there are a bunch of different loci that add together to produce personality. And that's not true either. In fact, genes are nodes in the same complex developmental process that the environment is. They're inputs into the environmental developmental process. It is possible sometimes to detect correlations across them, but the actual pathways are very hard to identify. Okay, we no longer take seriously the possibility that there are specific environments that confer specific risk for specific outcomes. But somehow, because of the technological gloss of the genome, we're tempted to think that way, right, for genes, that maybe there are specific genes with specific outcomes. I'm getting close to the end. In many ways, I think we've come full circle. I made this slide this morning and got defeated in getting a nice circle in PowerPoint. We started with twin studies, right? Twin studies showed that everything is heritable, summed tiny effects over many genes, but didn't turn out to be very good at identifying particular biological causes. So we then turned to candidate gene studies in the hopes of finding particular biological causes, but those didn't replicate because significance testing was such a failure in that context. And in response to that, we developed GWAS, which summed over those many tiny effects. But, well, I left out a step here with GCTA. I took those slides out. Well, anyways, we're back where we started. We've found out that heritability can sum over many tiny individual genetic effects, but that when we try and specify exactly what those tiny effects are, they dry up and blow away. So, coming back to my first slide, why don't I believe ultimately in the genomics of educational attainment? It's not because, and there are people who think this, so it's important for me to say it. It is not because I believe that educational attainment is somehow magically unrelated to our genetics. It's not. Yeah, I'm a behavior geneticist. I know that it is. It is related to genetics, as demonstrated by 100 years of twin and adoption studies. But this is not because there's a finite set of causal genes with effects that can be quantified. The result they report is essentially a report of a correlation of 0.014. And where in the history of science has an uncontrolled correlation accounting for, that is, 0.02% of the variance led down the road to a meaningful causal analysis? I don't think there's an example. So what that study is to me, it's a cool way to reconfirm that human differences are related to genetics, but we already knew that. A lot of the modern glossy genomics after candidate gene studies failed are new ways of showing us what we already knew, which is that in general, genetics is related to behavior. I think I'm going to leave time. That's a good place to stop. I think I'll stop right there and take some questions. So thank you very much.
Good. Thank you. So we have time for your comments and questions.
Yeah.
I have the crude impression that IQ was readily measured, and I think that's widely accepted to be true, and that IQ tended to track in families.
Yeah.
And I also had the impression, with quite a bit of personal experience, that autism could be understood and measured and separated, and also had quite a high probability of being genetically influenced. Now, that latter one I don't know anything about. I wonder if there's any information on that.
Yeah, the genetics of autism is very complicated because the twin studies were it's very hard to study because it's so, well, it's not as rare as it used to be, but it's still a very rare thing to study in twins. Because by the time you cross the prevalence of autism with the 1% of the population that is twins, it's very hard to find anybody to study. Autism does run in families for the usual kind of complex genetic reasons. The one thing I'll add, and I don't really have an opinion on it one way or the other, the interesting modern work on the genetics of autism is all about rare mutations. So you can now scan people's sequence, and everybody carries a mutation load. So you can scan people's sequence to find rare mutations, and the number of mutations that you carry is related to the probability of autism. And there are particular mutations that have been shown to occur in actually sibling pairs, not twin pairs, in members of sibling pairs who are autistic but not their co-twins. Frankly, I think that's probably just kind of the flavor of the month in autism genetics. I don't really think it's going to wind up explaining it because those same mutations are very highly related to IQ, for example. So it's in some ways a way of saying carrying a big mutation load is bad for you. And it's also related to schizophrenia. It's going to be like big ventricles or something. It's going to be related to everything bad. Yeah.
So, related question. You gave us a really nice summary of the frustrations in the world of gene identification, starting with linkage and candidate genes and so on. So now we have sequencing. And not only are people really excited about it in the genetics world, but also we're developing this big precision medicine initiative, and the hope is that we're going to transform clinical medicine. What do you think about that? That's my question.
Well, I mean, obviously it's easy to be skeptical, and not just. I'm a psychologist. If the question is about how useful is it going to be in ophthalmology, partly I just don't know. Except that I know. I think it's gone way slower than the best thing you can say is it's gone way slower than anybody expected. To me, in psychology and psychiatry, I think it's null. I don't think anything useful has happened. Would you disagree with that? There's not a drug you can point to. There's not an etiological hypothesis you can point to. They now have 68 SNPs in the PGC that are 10 to the minus 8, but all in all, put them all together, and it accounts for under 3% of the variance in the incidence of schizophrenia. And I don't see the results. And again, it's important for me to say that's not because I think schizophrenia is a strictly environmental thing. It's not that. It's that it doesn't analyze very well on a genetic level of analysis. I think the genetic effects on things like that are general, they're not specific. And I think people are getting, I guess there are some good results in cancer genetics, right? That drug development, but that's in the what do I know category. Yeah.
So it seems like trying to find individual genes or individual environmental factors that causally, mechanistically lead to behaviors or psychological traits is kind of too fraught because of everything you said. It's just almost infinite number of tiny little things interacting and adding up. So is there any hope for trying to scientifically study what causes people to be who they are, or is it just a completely hopeless concept?
you know, like I say, I'm a research psychologist, and I'm nevertheless